Let's take a look at how sexual reproduction maintains life's continuity while also introducing incredible genetic diversity. Sexually reproducing organisms like humans go through what's called the sexual cycle, a repeating pattern that alternates between diploid and haploid stages. In humans, our somatic or body cells are diploid. They have two sets of chromosomes, one from each parent. But to make gametes, egg and sperm cells, we can't just divide by mitosis because that would keep the chromosome number the same. Instead, we use a special type of cell division called meiosis. Meiosis reduces the chromosome number by half, producing haploid gametes that each carry one set of chromosomes, or one chromosome from each homologous pair. When a sperm fertilizes an egg, those two haploid cells fuse to form a diploid zygote, restoring the full chromosome number. From that zygote, mitosis takes over, creating all new cells of the growing organism. So, in short, meiosis creates gametes, fertilization restores the diploid state, and mitosis builds the multicellular body. Not all organisms use this strategy. Some reproduce asexually, producing offspring that are genetically identical to themselves. This method is fast and it doesn't require finding a mate. These are great advantages when conditions are stable and resources are plentiful. For asexual organisms like bacteria, whose lifespans are pretty short and cell turnover is really fast, enough genetic variation can be generated from mutation alone for these organisms to survive evolutionarily over what's been millions and millions of years. And actually, not all asexual organisms are microscopic or prokaryotic like bacteria. Take the hydra for an example. This is a tiny freshwater animal related to jellyfish and corals. Hydra are multicellular eukaryotes, meaning their cells have nuclei and organelles just like ours. Hydra can reproduce asexually through a process called budding. Basically, a small outgrowth forms on the parent's body. It develops tentacles and a mouth and eventually detaches as a genetically identical clone. This process is using mitosis, not meiosis, since no gametes or fertilization are involved. But sexual reproduction has a major evolutionary advantage, and that's increased genetic diversity. That variation helps populations adapt to changing environments, resist diseases, and avoid the buildup of harmful mutations over generations. When it comes to sexually reproducing organisms, enhanced diversity is the name of the game. It is the strength of our species and something that is to be celebrated and valued. So, where does genetic variation come from? In asexual reproducing organisms, variation definitely still occurs, but it's mostly due to random mutations in DNA. In sexual reproduction, we still have genetic variation through mutation. However, there's also variation built right into the process itself. So there's three key mechanisms to ensure that every individual is genetically unique, with the exception of identical twins. So we've got independent assortment, crossing over, and random fertilization. Let's start with the independent assortment of chromosomes. So during meiosis 1, homologous chromosome pairs, one set from each parent, line up along the middle of the cell during metaphase 1. Here's the key idea. Each pair aligns independently of all the others. This means the orientation of one pair doesn't influence how another pair lines up. For a simple example, imagine an organism with just two chromosome pairs. There are 4 possible ways to arrange them into gametes. Now, if we scale that up, because humans have 23 pairs of chromosomes, the possible combinations of maternal and paternal chromosomes that can be sorted into gametes is 2 to the 23rd. That's over 8 million unique possibilities of chromosomes for a gamete, and this is even before we talk about crossing over or random fertilization. So this process alone shuffles entire chromosomes, creating huge variation from one gamete to the next. And remember, when I'm talking about gametes, I mean eggs and sperm, which are the cells that are the product of meiosis. Next, we have crossing over. So independent assortment mixes up whole chromosomes, but crossing over goes even deeper. It mixes up genes within each chromosome. So during prophase 1, homologous chromosomes pair up side by side in a process called synapsis, forming these structures called tetrads. Okay, so at points along their length, the chromatids from each homolog or homologous pair physically exchange corresponding segments of DNA. This is called recombination. So when this happens, genes from the maternal and paternal chromosomes are swapped, creating recombinant chromosomes, new combinations of alleles that neither parent had. So, even if two gametes received the same set of chromosomes through independent assortment, however unlikely, crossing over ensures that the genes on those chromosomes aren't arranged in the same way. In humans, crossing over occurs 1 to 3 times per chromosome pair, so the potential for new combinations is just enormous. And finally, we have the concept of random fertilization. So, this adds yet another layer of variation. Each egg and sperm cell is already genetically distinct, thanks to independent assortment and crossing over. But which sperm actually fertilizes which egg is completely random when we're taking into account two different people. That means the genetic makeup of a zygote results from an unpredictable pairing of two unique gametes. If we combine the 8 million possible gamete combinations from each parent, the total number of possible zygotes is around 70 trillion. That's 8 million times 8 million. And that's even without considering the extra mixing from things like crossing over. So the combination of these three processes ensures that each new individual is truly one of a kind. So in summary, we've talked about a few sources of genetic variation in sexually reproducing organisms. We talked about independent assortment of chromosomes, which is basically shuffling chromosomes around, crossing over, which swaps pieces of chromosomes to create recombinants, random fertilization, bringing together two unique gametes. Together, these processes generate the extraordinary genetic diversity that fuels evolution and adaptation and ensures that no two individuals are exactly alike.